Today, we are going to see about the principle and process of biotechnology. In that, we are going to see about herbicide tolerant. Weeds. Weeds means what? Weeds means unwanted plant are a constant problem in crop fields. Weeds not only compete with crops for sunlight, water, nutrients, and space, but also a carrier for insects and disease. If left uncontrolled, weeds can reduce crop yield significantly. Glycophosphate herbicide, and this is produced by Monsanto USA company under the trade name Roundup, kills plant by blocking 5-enopyruvate sikimate 3 phosphate synthase. It is called as EPSPS enzymes, an enzyme involved in the biosynthesis of aromatic amino acids, vitamins, and many secondary plant metabolites. There are several ways by which crop can be modified to be glycophosphate tolerant. One strategy is to incorporate a soil bacterium gene that produce a glycophosphate tolerant from form of EPSPS. Another way is to incorporate a different soil bacterium gene that produces a glycophosphate degrading enzymes. Now we are going to see about the advantages of the herbicide tolerant crop. Weed controls improves the higher crop yield. Weed controls and improves the higher crop yields. Second one, it reduces the spray of herbicides. Third, it reduces the competition between crop plant and weed. Next, use of low toxicity compounds which do not remain active in the soil. Next, the ability to conserve soil structure and microbes. Herbicide tolerant Basta. The trade name Basta refers to a non-selective herbicide containing the chemical compound phosphophynoerythricin. Basta herbicide tolerant gene PPT was isolated from Medicago sativa plant. It inhibits the enzyme glutamine synthase, which is involved in amino acylation. The PPT gene was introduced into tobacco, and transgenic tobacco produced was resistant to PPT. Similar enzyme was also isolated from Streptomyces hydroscopix with bar gene encodes for PAT, that is phosphophynoerythricin acetyltransferase, and was introduced into the crop plant like potato and sugar beet, and transgenic crops have been developed. Insects resistance BT crop. BT cotton is generally modified organism or genetically modified pest resistant plant cotton variety which produces an insecticide activity to bollworm. Strains of the bacterium Bacillus genus produces over 200 different Bt toxins, each harmful to different insects. Most Bt toxins are insecticidal to the larvae of the moths and butterflies, beetles, cotton bollworms, and gadflies, but are harmless to other forms of life. The gene are encoded for toxic crystals in the cry group of endotoxins. When insects attack and eat the cotton plant, the cry toxins are dissolved in the insect's stomach. The epithelial membrane of the gut block certain vital nutrients, thereby sufficient regulation of the potassium ions are lost in the insects and result in the death of the epithelial cells in the intestine membrane, which leads to the death of the larva. Now we are going to see about the advantages. The advantages of BT cotton are: yield of cotton is increased due to effective control of bollworms, reduction in insecticide use in the cultivation of BT cotton, potential reduction in the cost of cultivation. Now we are going to see about the disadvantages. BT cotton has some limitations. Cost of BT cotton seed is high. Effectiveness up to 120 days. After that, efficiency is reduced. Ineffective against sucking pests like justice and aphids and whitefly. Affects pollinating insects and thus yield also is affected. BT brinjal. The BT brinjal is another transgenic brinjal created by inserting a crystal protein gene from the soil bacterium Bacillus thuringiensis into the genome of various brinjal cultivars. The insertion of the gene along with other genetic elements such as promoters, terminators, and an antibiotic resistant marker gene into the brinjal plant is accomplished using agrobacterium mediated genetic transformation. The BT brinjal has been developed to give resistance against lepidotron insects, in particular the brinjal fruits and shoot borer. Next, we are going to see about Dara mustard hybrid. DMH11 is the transgenic mustard developed by a team of scientists, Center for Genetic Manipulation of Crop Plants at Delhi University, and a government sponsored project. It is genetically modified variety of herbicide tolerant. It was created by using Barney's Barstar technology for genetic modification by adding gene from soil bacteria that make mustard a self-pollinating plant. DMH11 contain three genes: bar gene, Barney's, and bar stars sourced from the soil bacterium. The bar gene had made plant resistant to herbicide named Basta. Virus resistance. Many plants are affected by virus attack, resulting in serious loss in the yield and even death. Biotechnologically, invention is used to introduce viral resistant gene into the host plant so that they can resist the attack by virus. This is by introducing genes that produce resistant enzymes which can deactivate viral DNA. Flavor sour tomato. Agrobacterium mediated genetic engineering technique was followed to produce flavor sour tomato, that is, retaining the natural color and flavor of the tomato. Through genetic engineering, the ripening process of the tomato is solved down and thus prevent it from softening and to increase the shelf life. The tomato was made more resistant to rotting by agrobacterium mediated gene transfer mechanism of introducing an antisense gene which interferes with the production of enzyme polygalacturonase which help in delaying the ripening process of tomato during long storage and transportation. Golden rice biofortification. Golden rice is a variety of Oryza sativa produced through genetic engineering of biosynthesized beta carotene a precursor of vitamin A, in the edible part of the rice developed by Indigo Porticus and his group. The aim is to produce a fortified food to be grown and consumed in the areas with a shortage of dietary vitamin A, which kills so many children under 5 years of age. Golden rice differs from its parental strain by addition of 3 beta carotene biosynthesis gene, namely Xii, that is phytin synthesis from daffodil plants, and CRT1 gene from the soil bacterium Erwinia aerodura, and LYC, that is lycopene cyclase gene from wild type rice endosperm. The endosperm of normal rice do not contain beta carotene. Golden rice have been genetically altered so that the endosperm now accumulates beta carotene. This has been done using recombinant DNA technology. Golden rice can control childhood blindness, that is xerophthalmia. Genetically modified food benefits: high yield without pest and 70% reduction of the pesticide usage, reduce soil pollution problem, conserve microbial population in soil. Risk factors that is seen in the genetically modified food: affects liver, kidney function and cancer, hormonal imbalance and physical disorder, anaphylactic shock and allergies. Anaphylactic shock means sudden hypersensitive reaction. Adverse effect in immune system because of bacterial protein. Loss of viability of seeds shown in terminator seeds technology of GM crops. PHB means polyhydroxybutyrate. Synthetic polymers are now biodegradable and pollute the soil, and when burnt add ditoxin in the environment which cause cancer. So, efforts were taken to provide an alternative eco-friendly biopolymers. Polyhydroxyalkanoates and polyhydroxybutyrate are group of degradable biopolymers which have several medical applications such as drug delivery, scaffold and heart valves. PHAs are biological macromolecules and thermoplastics which are biodegradable and biocompatible. Several microorganisms have been utilized to produce different types of PHA, including gram-positive like Bacillus megaterium, Bacillus subtilis, and Corynebacterium glucominicum. Gram-negative bacterium like group of Pseudomonas species and Alclogens eutrophus.